Mike Dolinsky here with Taurus.ag. So we'll take a quick look at the label for PLS so you know what nutrients are in there because I'm going to be talking about them. And then we're going to look at nodule formation and the fixation process, and then factors affecting end fixation and nodulation itself. But first, let's talk about seeds. We know from past experience that seeds have different levels of nutrient in them. In this particular sample, you can see from our plots, you got small green seeds that were a little later in developing, probably in the top pods. You got these huge, monstrous big seeds, which were probably formed early. And there's going to be different levels and different mixes of nutrients in these seeds. So they're going to be a little bit different. So one of the things we do with things like active prime on cereals and canola and other crops is we put a nutrient package right on the seed. That way it gives each seed an opportunity to secure certain nutrients. And there are specific ones necessary for nitrogen fixation, which I'm going to be talking about. But we also need some of those nutrients to grow a good seedling. You know, these products will enhance that productivity until the roots can get going. So here's a look at some pea plants, and you can see that this one is germinated and the shoot is going up, and we got these lateral roots that are developing. So if there's any nutrient right on the seed and it's soluble, it can be absorbed, or these roots will pick it up very quickly after germination. And then very quickly you can see how much the lateral roots develop on that primary main root. And here's a shot showing the root hairs, and this is a close-up basically of this particular shot. You can see that the root hairs are very plentiful on peas, and they are extracting nutrients from this granule. About 60% of all the nutrients are taken up by root hairs, and they're only single cells, so they live 5, 6, 7 days, and if the root isn't growing, then the plant has trouble accessing those immobile nutrients. But that's another story for another day. So there's a lot of root hairs, and this is where infection by the rhizobium takes place. So let's take a look at PLS, and what PLS stands for is peas, lentils, and soybeans. You can put it on faba beans and chickpeas as well. So it'll support rhizobium and bradyrhizobium inoculants as seen here. And the treatment dosage is about 2 mils per kilogram of seed, which doesn't seem like a lot. But with it right there on the seed and right near those roots as soon as the plant germinates, it's fairly concentrated right in that small zone, and the plants can extract that quite nicely. So it's 2% nitrogen, 10% phosphate, and 10% potash. So this helps the entire plant as well as the nodules themselves, and have a role in terms of fixation, which we'll get to later. But it's got 0.1% boron, some cobalt, iron, manganese, molybdenum, nickel, and zinc. These transition metals are needed for all the reaction processes that take place and catalysts in enzymes. So that's the sort of spectrum I'll be covering here throughout this presentation. Now let's take a look at how a nodule forms. The first thing that happens is the microbe, the bacteria, has cilia on them, or little swimming paddles, so it can swim over to a nodule, and it's attracted there by flavonoids, which are given off by the root. So it can find it, and these organisms, the rhizobium symbiotic relationship, has been going on for about 65 million years, so they've got a lot of experience. In mycorrhizae, that relationship goes all the way back to the beginning of plants, about 450 million years ago. So we'll leave it at that, but take a look at this. So when the microbe gets there, it signals the plant, which are just turned node factors, so that the root hair knows that the microbe is around, and then it attaches to the root tip, or the root hair tip. And then the plant, because it's signaled, has a calcium spike, which brings about a change in terms of the genetic upregulation of node factors. So the plant is actually prepared for that and curls around that bacteria and forms what's called an infection thread. So that infection thread grows down into what's called the pericycle down here near the vascular root system, and then the plant builds the nodule. And the plant builds the nodule, and it sort of restructures the root surface, and it uses a lot of calcium and boron. So during that period when this restructuring is going on and the plant is both building and also starting up fixation, the plant can almost stall because it's using so much energy from the shoot to feed the root that there's not enough left to do much growth of the shoot. The root can't feed itself, so all that carbon and the sugars that are sent down are really the basis of the energy that is needed to form all those cells and support the development and the growth of these bacteria. Because it's the bacteria that want those sugars in exchange for the nitrogen that is going to fix. And the same kind of spike from calcium occurs here in mycorrhizae as well. So it's believed that the rhizobium captured that process based on what the mycorrhizal had developed over hundreds of millions of years. Kind of an interesting story, that's all. So what is that fixation worth? Well, you look at chickpeas, 135 pounds an acre, soybeans about 100 pounds, lentils about 80 pounds, alfalfa lots, but in perennials you look at, or annuals you look at peas at about 150 pounds and faba beans about 200 pounds. I mean, faba beans is a fabulous fixer, and legumes as a whole have small roots, so they've sort of evolved using the symbiotic relationship to compensate for their poor root system. Now here you can see that this root hair has had an infection, and the root hair is curled around the bacteria. So here's what we have. We have two kinds of nodules, and this one is specifically in what are called the tropical types or determinate nodules. And these are what we generally have here in most of our legumes, indeterminate nodules. And this just shows where the fixing is taking place. This is sort of getting old, and this is going to be new fixing. It sort of progresses over time, and the nodules are a bit longer. So here's an indeterminate nodular. This is in peas, and you can see here what I was talking about. This is fixing nitrogen. This is sort of aged, and this is going to be new material. When the plant and the nodule have finished their relationship and it's worn out and getting old, here's what's left. You can see that this is an old nodule, and all there is left there is just a tiny bit of fixing going on there. This is going to soon die and shrivel up and disappear into dust in the soil. Now here are some other shots showing the same kind of thing. This one here, you can see this process in an indeterminate nodule, fixing, not fixing. Faba beans, the same thing. I just threw these in because lentils have very small nodules, and it's amazing that can produce up to 80 pounds of nitrogen per acre, but it seems to be a very, very active fixer. It's really bright red, and this bright red is a sign that fixing is taking place, and the redness is from a compound called leg hemoglobin, similar to human hemoglobin, in that it regulates oxygen in the nodule. And I'll tell you why in a minute, but those are the kinds of indeterminate nodules we have in pretty well all of our legumes and pulses except for soybeans. This is a soybean nodule, and you can see it's different. It's nice and round. And here's a shot of a whole bunch of nodules. I grew these in my backyard here in Edmonton, so we know we can get nodulation of soybeans even up here in this climate. So here's the process going on here. So this nodule in soybeans is very small. You can see there's no red, so there's no active fixing going on. This one is small, but it's starting up. And here you can see one that is in full tilt, and it's basically hooked right to the xylem phloem, the vascular system here. So as it fixes nitrogen, it can pump that nitrogen into the xylem and move it up, and it can pull in the sugars and the carbon that it needs here for energy coming down the phloem. And that's basically what happens. So you can well imagine as a plant is developing all of these nodules and starting it up, it takes a horrendous amount of energy to do that, and it has to be coordinated. The fact of the matter is that this is so energy dependent that if you have too much nitrogen there, the plant won't even bother trying to fix nitrogen because it's a lot easier for it to actually take nitrates and ammonium from the soil than it is to build this nodule. So high levels of nitrate especially will interfere with this. Now here's the actual process. So we have this molecule N2 in the air, which is the nitrogen that we breathe in all the time, and it's 78% of the atmosphere all the time. It's very, very stable. Now, the only way we can fix nitrogen, there's sort of the Haber-Bosch process, which is not a biological process, but a chemical reaction process, which feeds a big chunk of the world with the production of urea. But most of the nitrogen fixed from the atmosphere is done by nitrogen-fixing organisms, Rhizobium, and there's a whole bunch of other ones that nobody much talks about in various kinds of plants. So that energy, the energy that is needed is really intense. In fact, the other way that we get fixation of nitrogen is through lightning. In a lightning storm, and there's about 3 million lightning strikes a day in the world, the energy from lightning can break this triple bond in one shot. And then as it's raining, those ends can hook up with oxygen and perhaps hydrogens in the atmosphere, and you would get nitrate, and we can actually get some ammonia and maybe even ammonium coming down, and we get a nice flush of nitrogen in the crop response after a rainstorm, and that's why that happens. And here's the process, and it takes a whole lot of ATP, and I'll cover that a little more in detail in a sec. So it takes a lot of energy to break one of those molecules. Now, nodules act as a strong sink for moly and phosphorus. And it's such a strong sink for phosphorus that it will push the phosphorus into the nodule if it's short at the expense of the shoot. And moly, it's a strong sink because without molybdenum, the whole process can't take place, because molybdenum is in fact the catalyst, the transition metal that is used to break that triple bond, one bond at a time. And here we can see the molecule. This is nitrogenase. You can see it's got molybdenum, a lot of iron, and a lot of sulfur in that molecule. So we have dinitrogenase reductase, so it's got to be reduced a bit before it is then fixed or converted from N2 to H3. It's basically the same as the process of breaking down urea to ammonia. You get two ions, molecules of ammonia. It picks up another hydrogen and becomes ammonia very quickly, because ammonia is toxic to the plant. And you can see there's that ATP process in here using up a whole pile of energy. Now, before you get scared looking at this, it's not as complicated as you would think, and it really summarizes the whole process. Here is sugar coming down from the shoot, from photosynthesis. So you need a really good plant that's producing a lot of sugar to have a really active fixation process. So you get the sugar coming down, it's taken into the cell as malate. It goes into the citric cycle or the TCA cycle, and what we get out as ATP. That's where the ATP is generated. Okay, so we're starting there. We get the ATP hooked into N2, nitrogen from the atmosphere, with that nitrogenase complex. And you'll notice there's hydrogen here being pumped in to the actual membrane, inside the membrane of the symbiosome of the bacteria. So we get that converting to ammonia, and then that is converted, and it picks up a hydrogen here to ammonium, because the ammonia itself will be toxic. And this process, the GS GOGAT system, is the way that plants, as well as basically all plants, take in nitrogen and get it into amino acids. So we have ammonium, and then we get glutamate, which is then part of that process, and we get glutamine, the amino acid glutamine, and we can have asparagine as well produced from glutamine, and those two amino acids are the transporters of nitrogen in the system. So you can see that those move into the xylem and then they can be moved up into the shoot. The plant will break them down in the cells and form all the amino acids along with, you need sulfur in there because not just nitrogen in terms of forming those amino acids. One thing the soybean growers need to know is that in soybeans, the conversion is into something called urease rather than in terms of these two amino acids. And it is converted to amino acids up in the shoot. And it's just specific to basically those determinant nodules. And nickel is really key in that process of breaking those ureases down into glutamine and asparagine. And one of the other things I want to show you here is you take a look at in the membrane, we have transporters of sulfate, molybdenum, iron, magnesium, and a whole bunch of others that aren't showing. So the plant has these transporters to make sure it gets that molybdenum, it gets that iron, it gets that sulfur, those key things that are required for synthesis of your ammonia, your fixation. It's an amazing relationship that has developed over that 65 million years. The nutrients that are essential for fixation, talked about most of these. Calcium for signaling and the cell restructuring. I don't have to say anything more about P. If you're short of P and you're really short of P, it's something you should consider supplementing your crops with, all crops basically. And that's why we have a fair amount of phosphorus in the PLS product. Iron for electron transport, sulfur for the iron linkages with sulfur in there, and also the sulfur is needed for making amino acids within the actual bacterial organism itself. Boron for restructuring cells. Nitrogenase for the process of actually breaking those bonds. Leg hemoglobin, which contains cobalt, one of the few places you'll find a role for cobalt, is used to regulate the oxygen going to the nitrogenase. The trick is nitrogenase cannot function at high oxygen levels, like atmospheric oxygen levels. So between the actual membrane around the symbiosome that restricts oxygen in there, the leg hemoglobin also downregulates oxygen supply so the nitrogenase will function, and it also is used in vitamin B12 production. Nickel is now one of the essential nutrients that are required in plants, and it's involved in urease, the enzyme that breaks down urea, and there's some urea that gets sorted out in the plant, and it needs to break it down into ammonia and then ammonium. It also is involved in the molecule or the enzyme hydrogenase, which is used in the plant to deal with hydrogen, because hydrogen ions can become toxic, so they have to be broken down. And it's now known to be involved in reducing stress in plants as a whole and in the nitrogen fixation process. It also has zinc in it, and zinc is used in enzyme activity of all kinds. It's key to all enzymes. It's in all the enzyme groups, and it's involved in reactive oxygen species scavenging. So those are the key nutrients. One thing to point out here is molybdenum is kind of a unique in that as the pH drops, the availability of molybdenum decreases. So if you got acid soils, that would be the key place where you would look for maybe supplementing additional molybdenum beyond what is in the seed dressing. With nickel sort of responds about the same as copper and zinc as the pH goes up. In fact, nickel as a divalent ion becomes less available starting about pH 7 from what I can tell in the literature, so it's more available in acid soils. The main factors limiting biological nitrogen fixation: too much or too little water, drought, and excess water. Both of them are bad for you. Soil acidity and alkalinity, anything below about 6.5, depending on which organism, and anything over about 7.5 can be deleterious to nitrogen fixation. And salinity is always bad for microbes and for seeds and everything else. The amount of sugar being supplied by photosynthesis is key to driving it. P deficiency can be a real hamper for you. High temperatures can be dangerous to microbes like that, especially especially before they get established. Toxins and heavy metal. Excess mineral N, I talked briefly about that earlier, but yeah, if you have much more than about 25, 35 pounds of N available in the soil, uh, you might start interfering with nitrogen fixation because, as I said, it's cheaper for the plant to just take it from the soil. And that's one of the key questions people ask. Back a couple of years ago, we had a drought and there was a lot of nitrogen left over at the end of the year. And when you get that kind of situation and you do a soil test and you find, 50, 60 pounds of N, might not be a good field to put a nitrogen fixing crop in. And the other thing is slow transport and metabolism of the nitrogen that is fixed. If the plant can't move that nitrogen away from the nodules, the plant and the nodule will downregulate production. They recognize they're overloaded. So you got to be able to have that plant growing actively. And then all these deficiencies are really, the end of the picture. So thanks a lot, and always remember that the plant knows what to do, and this relationship's been going on for millions of years. All we got to do is figure out how to help it. Well, anyhow, good luck and have a great season.